I'm gonna add a little bit more kind of basic research information about phages and arms race with Vibrio cholera to what Kim just talked about. And then for the second part of my talk, tell you about how we're using these phages that Kim presented to try and prevent cholera. So not quite phage therapy, but phage prophylaxis. So Kim gave a good introduction, and I can probably skip almost all this. I'll say a little bit about the organism, a little more. So there are 150 different LPS O-antigen types out there that gives rise to the serogroups. But most cholera in the previous pandemics and in the current pandemic is caused by this O1 serogroup, which makes it kind of an easy target if you're using phages, because the O-antigen is a major receptor for phages amongst the gram-negatives. And so this is just a little schematic for those of you that are not familiar with gram-negatives. They have two membranes, an inner and an outer. And so the outer leaflet of the outer membrane is the lipopolysaccharide. And this is just a schematic of this O-antigen sticking off of the cell. So why do we still have cholera outbreaks? There are vaccines, whole cell killed vaccines, live attenuated vaccines, and they give partial, short-lived protection, which is good, but it's not nearly as good as if you survive a bout of cholera, where you have high-level, long-lasting immunity that lasts greater than 7 years. And so these vaccines are being used, but mostly on a clinical trial basis. And it's a problem that we've heard about in previous talks, is cholera strikes mostly in poor areas of the world that can't really afford widespread vaccination. So that continues to be a problem. And so this O1 antigen that's going to crop up in my talk a couple times is about 12 to 18 repeats of a pyrosamine backbone with tetranate modifications coming off of it. And I'll get back to that in a second. So this is the result of cholera infection. Once the fluids pass through and cleared out the contents of the intestinal tract, patients just shed liters and liters of this, what's called rice water stool. And as Kim said, this is loaded with Vibrio cholera that go and transmit back into the environment. And Felix Durrell 100 years ago discovered phages in cholera patient rice water stools and for other pathogens as well. So Kim also mentioned this, but I'll go into a little bit more detail. So kind of the traditional view of the life cycle of how I call it toxigenic Vibrio cholera. I should mention that most Vibrio cholera in the world don't cause cholera. They don't have the cholera toxin genes. They don't have a lot of the virulence factors. So we call it the toxigenic Vibrio choleras that cause epidemics and pandemics. So the traditional view of the life cycle is a susceptible individual drinks contaminated water or food, they get cholera, and out comes the bacteria by the billions, contaminating the water further. But it's become more and more appreciated that a major other mode of transmission is this rapid mode of transmission that happens within households. And some recent studies have estimated that the rate of infection goes up about 2 orders of magnitude if you reside in a household where somebody comes down with cholera. And the peak incidence of these secondary cases is about 2 to 3 days after the index case. So this presents a huge problem in trying to control cholera and preventing and stopping outbreaks. Because there's not enough time to vaccinate individuals, these household contacts. And although antibiotic prophylaxis would be efficacious, it's not recommended because of antibiotic resistance. And so I want you to keep in mind this household transmission, because we're thinking perhaps we could design a cocktail of phages that we could give to the household contacts to protect them. And so in thinking about prevention or prophylaxis using phages for cholera, there's a number of questions that we've asked First of all, are there Vibrio cholera specific lytic phages that are virulent in the host small intestine? And as Kim said, Felix Durrell basically discovered this 100 years ago. So the answer is yes. What's the biology of these phages? What are the receptors? Are they, how virulent are they? Do they form lysogens or prophages? And of course, Vibrio will become resistant to these, and we would like to know what are the mechanisms of escape from these phages. And importantly for phage therapy or prophylaxis, do escape mutants remain infectious? And then finally, can single phages or cocktail prevent cholera? And so I'll present data that addresses all these questions. Oh, and then the final thing we've recently done is to make it, we have Vibrio cholera specific phages, but we went on to show that these phages don't disrupt the gut microbiota. So our work on phages started many years back by a former MD PhD student, Eric Nelson, who did his PhD work in my lab. And he was in Bangladesh isolating stool samples at the ICDDRB, and half the samples had phages, and he had to throw them away because you couldn't do anything with them. And he kept emailing me and saying, we should be studying these phages. And I said, okay, let's study these phages. And so he did this simple experiment that had never been reported before, which is if you just give the phage in the inoculum, can it prey on Vibrio cholera in the small intestine in an animal model and reduce the load? And so we use this, the most common model in the cholera field is the infant mouse model. And sure enough, if you increase the amount of phage added along with the inoculum, you can reduce the load of Vibrio cholera. Unbeknownst to us, Shah Farooq's lab was doing similar stuff and published, we published in the same year, which was nice. So this answered showed evidence of what Felix Durrell had hypothesized a century before, that Vibrio-specific phages that we find in stool samples are virulent in the context of, at least in this case, the infant mouse small intestine. So picking up on what Kim talked about, through this collaboration with Firdousi Qadri, who's a microbiologist / immunologist at the ICDRB, we took advantage of her 10-year collection of rice water stools and found these phages, ICP 1, 2, and 3. And this was either by finding plaques directly on a lawn from the stool sample, or once we had identified the phages and sequenced them, Kim went back and did PCR. And so you can see ICP1 was omnipresent throughout all these years and still is to this day. And then these other two phages are a little bit more sporadic. And so I agree with what Kim said, that this, in a way, is shocking, because you can go out to estuaries or wherever you can find Vibrio cholera and find lots of phages. There's been hundreds of Vibrio-specific phages that have been found and used for typing. But year in and year out, there's only these three phages that we find. And so we have hypothesized that these phages have evolved to take advantage of these cholera outbreaks. Tag along with Vibrio cholera, because if you do that, and if you have what it takes to survive in the human intestine, then you just wait, and the bacteria are going to grow to high titer, and then the phage can then bloom on these bacteria. And so we think these phages have evolved to do this. And I'll show you some data that tell you a little bit more about these phages and how they're successful. So here's the three phages that Kim presented. Just for those that don't really know what these families mean. So T4 is probably the most well-studied myoviridae, and T7 is a podoviridae. And we've recently started to generate three-dimensional images of these using cryo-electron tomography. This is in collaboration with Elizabeth Wright at Emory. And they're all short tail fiber type phages. And then receptors. So the receptor for ICP1, and we have some evidence showing that it's the receptor for ICP3 as well, is this O1 antigen. So this is kind of good news, since most cholera is caused by O1 serogroup strains. And this O1 serogroup doesn't appear to be going away, despite it being the dominant antigen for immunity. This other phage, ICP2, uses a different receptor. It's a protein in the outer membrane called OMP-U, and I'll tell you a bit about that. And so Kim, when she was a postdoc at my lab, started to characterize these phages and the receptors. And the story for ICP1, this omnipresent phage, was very interesting, because if you put the phage together with Vibrio cholera in a flask, very quickly Vibrio spits off these escape mutants. And what Kim showed using whole genome sequencing is that within the biosynthetic operon for the O1 antigen, there are two genes, WBEL and MAN-A, that have these poly-A tracks. And what she showed is that at a high frequency, through slip-strand mispairing, they will add or subtract an A and shift the reading frame of the gene. So it's not making the protein product, and then the bacteria are not making the O1 antigen, and they escape the phage. But we don't see these frame shift mutants coming out of cholera patients. Why is that? It's because the O1 antigen is required for infection. It's a virulence factor. That was shown by Matt Waldor and John Mekalanos back in the 90s. And so Kim went ahead and took these frame shift mutants and showed that indeed they are attenuated. This is the competitive index competing against the wild type in our infant mouse model. So this is the WBEL frame shift mutant, and here's the MAN-A mutant. And you can isolate revertants that restore the reading frame of the gene, and then they regain virulence and can compete 1-to-1. So this is kind of the first result I wanted to get across to you, is that one of the reasons we think ICP1 is so successful is it's targeting a receptor that's a critical virulence factor of the bacteria. And so the bacteria can't just get rid of that, turn the genes off, or mutate it during infection. And that's a recurrent theme, as you'll see. So Kim just gave a great talk on the plea, so I won't go into this, but there is an arms race going on here, and so Vibrio has this defense mechanism against ICP1, and yet ICP1 is omnipresent in Bangladesh and probably in other areas. And as Kim talked about, the phage can overcome this with this CRISPR-Cas system. And so just to go on to a little bit more detail, and Kim showed this in our Nature paper, so all of these spacers in this green color here target protospacers in one or more of these different pleas. And so for example, this 2011 isolate, spacer 8 and 9, target these two genes here. And Kim showed if you destroy this targeting, you can get rare plaques where the phage has now picked up a new spacer that targets another gene, another protospacer in the plea. So this is a fully functional CRISPR-Cas system that the phage has. So somewhat luckily, this 2011 phage has spacers targeting all of the pleas that are in current circulation. And so this is the first component of this phage cocktail that I'm going to tell you about in a minute that we're using for prophylaxis. So let me say a bit about ICP2. So that's a different type of phage. We find it sporadically, and it's a different member, a different family member, and it uses a different receptor on the surface, and that is a protein called OMPU. So we published this a few years ago, and Kim, this is mostly work from Kim's hands, is that we would find in cholera patient stool samples where you would find Vibrio cholera and ICP2, you could find escape mutants. And so this, by sequencing these escape mutants, we quickly figured out what the receptor is for this phage. Some patients were shedding ICP2 escape mutants that had null mutations in a gene called TOXR. And this just showed there were some stop codons, there were some amino acid changes in critical residues. All of these abrogate function of this protein. So why, this is a transcriptional regulator. Why would this give resistance to this phage? Well, TOXR has been studied for many years, mostly by John Mekalanos' laboratory, but also some other laboratories. And what's been shown is it's an intermembrane protein that has a DNA binding motif that is a transcriptional activator of genes. During infection, this TOXR protein is expressed and regulates virulence genes. It also is a positive regulator of OMPU. And so knocking out TOXR function lowers OMPU expression, and then the bacteria can escape the phage. But as I just mentioned, TOXR is a major virulence gene regulator. It's a positive regulator of the cholera toxin genes, a pilus that Vibrio cholera needs to colonize. And so these patients, due to phage pressure, are actually shedding avirulent mutants out so that they can escape the phage. And Kim went ahead and showed that indeed these point mutants that are coming out of some cholera patients are highly attenuated. This is, again, a competition assay in the infant mouse model. So here's again another example of why we think these three phages are so successful, is they're targeting receptors that are critical virulence factors. So in this case, Vibrio is even willing to give up its virulence to escape this phage, presumably due to a bloom of the phage in these patients and a tremendous selective pressure. But Vibrio can fight back in another way. It's a little bit harder to get mathematically, but Kim showed this, that some patients are shedding isogenic mutants of Vibrio cholera that have these point mutations in these two outer loops of OMP-U. So this is a outer membrane porin, and this is a predicted structure based just on a FIRE2 prediction. And so all these mutations map to these two loops, and we assume that that is where the tail fibers of ICP2 engage to infect the bacteria. So in this case, Kim showed that these point mutations don't abrogate function of OMP-U. And it turns out this is important because OMP-U is a critical virulence factor. Vibrio cholera switches porins during infection because this OMP-U porin is less permeable to defensins, bile salts, and other antimicrobial molecules that the host has. So recently I have a graduate student who started to ask this question, does ICP2 tail fiber engage OMP-U, and in particular these loops? And so what she's been doing is taking some of these point mutants and then infecting with phage, a high number of phage, and getting host range mutants of the phage that can now plaque on these mutants. And this is Andrea Wong's work. And so this is just the genome of ICP2, and what she's found is if by taking, for example, a particular mutant of OMP-U and looking for host range mutants that can now plaque on it, she's getting mutations in these two genes, gene 23 and gene 25. And there's kind of trace homology for gene 25 to tail fibers of other phages, and so this wasn't that unexpected. Now, when she first gets these mutants that can form plaques, they're very turbid plaques, and they're small. And upon additional passaging, she'll get additional mutations that then give a nice virulent phage that can form clear plaques and multiply rapidly on this. But it takes a lot of mutations in the phage to do this. And she's been playing this game with a number of different OMP-U point mutants. And satisfyingly, all the mutations are occurring in these three genes. And so the phage can overcome these OMP-U mutations. And so why are we seeing multiple genes being hit? So the most well-studied member of the Podiviridae is T7, and there's been a lot of work from many decades of work showing that there's multiple interactions between proteins in the tail and receptors on the surface. And so we think that ICP2 probably also is interacting with multiple receptors on the surface for its infection. Okay, so now, so we've been characterizing kind of the basic biology of these phages, and we're starting to learn about why they're so successful for infecting, tagging along with Vibrio cholera and preying on Vibrio cholera in the human intestine. But the fact is that we're getting these phages from very sick people who come to the hospital with cholera. So they're not protecting these patients. So it's a little bit of a sample bias problem, because there's been several epidemiological studies that have shown that for every symptomatic individual that has cholera, there's probably 2 to 4 mild or asymptomatic cases. And you can see that in a lot of these household studies where people either seroconvert or are shedding Vibrio but have no symptoms. And so we think it's possible that the phages may actually be playing a protective role. But there's a sample bias problem in that they're not sick and we don't study them or analyze them. But the problem's worse than that, because phage therapy has been tried quite a bit for Vibrio cholera. And back in the 20s, 30s, and 40s, there were many papers published on successful phage therapy trials for Vibrio cholera, field trials. But in the modern era with well-done controls, it's not been such a rosy picture. So there was a paper published in 1971 where a multi-nation collaboration taking 11 Vibrio-specific, highly virulent phages and going and doing a clinical trial, and they were not effective. So this kind of put the nail in the coffin on phage therapy for cholera. And really, when people are sick, it's rehydration therapy and antibiotics to shorten the course of the infection. You don't need phages. They're already sick. The phages aren't going to help. But what about phage prophylaxis? There's been a few papers on this, mostly from the Sarkar lab in India, and they had a paper in 2013 that didn't bode well for phage prophylaxis. They were using an adult rabbit model of cholera, and it did not protect the animals, and they saw that the titers of the phage dropped 100-fold after just a few hours in the intestinal tract. But we're not daunted by these studies, and I think by finding phages that we know are virulent in the context of human infection, for some of the reasons I told you, we're hoping that using the right phages may work. And so we started doing work in animal models, and this is work that's been spearheaded by Mimi Yen and Lynn Cairns, who are two postdocs in my laboratory. So I'm going to show you some data from the infant mouse model for cholera. There's a problem with the infant mouse model in that they don't get profuse watery diarrhea like humans do. But there's another animal model, which is an infant rabbit model, that do get profuse diarrhea. So I'm going to show you data from both of these animal models. So in a typical experiment, this is in the infant mice, we'll dose the mice with phages, so about 10 to the 7th phage in this experiment. And I say minus 3 hours, so we'll then wait 3 hours and then challenge these mice with what would normally be a lethal dose of Vibrio cholera. We'll let the infection go for 24 hours and look at the load of the bacteria in the small intestine. And so what you can see here is if you don't give any phage prophylaxis, all the mice are highly infected, but using these individual phages that I've talked about, we see varying levels of protection. ICP1, this omnipresent phage, actually performed the worst. It protected a couple orders of magnitude, but almost all the animals were colonized, and we see lots of escape mutants, by the way, in these single phage prophylaxed animals. ICP2 and 3 worked better, especially ICP3, which is the most virulent of our phages. And then the cocktail worked very well as well. And we kind of predicted that since the 3 phages work differently, are targeting two different receptors. I won't belabor this point, but in these same animals, we can look at the concentration of phages in the small intestine. And what we see is that the population of phage generally correlates with the population of the Vibrios. So this is ICP1 treated, so there's quite a high load of bacteria. There's a high load of phages, and it correlates. And this is because the phage replication depends upon the population density of the host bacteria. So encouraged by that success with a cocktail of phages, we then started to study this further. How well retained are these phages? That was just a 3-hour prophylaxis. And so we got permission to go out to 24 hours in our infant mouse model. And what we are looking at here is retention of the 3 phages over time. And what you can see that the phages are very well retained. By 24 hours, there's still good retention of ICP1 and 2, but ICP3 is really starting to wane. So we've looked at, we're now going to test prophylaxis out to 24 hours. So this again is the infant mouse model. If we don't give any phage, all of the mice are colonized at a high level. And at 6 and 12 hours, we see this bimodal protection where many of the animals clear the infection, but some of the animals are colonized, albeit several orders of magnitude lower than when you don't give phage. We looked very carefully at the Vibrio cholera that were present in the 6 and the 12-hour prophylaxis groups, and we do not see escape mutants coming out. However, in the 24-hour prophylaxis experiment, so all of the animals are colonized, but a couple orders of magnitude less than no phage prophylaxis. But we do see lots of escape mutants in these animals. However, we don't see escape mutants against all 3 phages. So now I'm going to switch to the infant rabbit model, which They get colonized, but then they get profuse secretory diarrhea just like humans. So here, this is a much larger animal model, so we give a lot more phage, and we give a large dose of Vibrio cholera required to cause disease in them. So if you don't give phage prophylaxis, all of these animals are colonized to very high levels, and they're all very sick, and we actually have to euthanize them very early in the experiment because as soon as they lose 10% of their body weight, we euthanize them. So the 3 and the 24-hour prophylaxis groups are shown here. The 3-hour gave this, again, this bimodal protection. 4 of the animals cleared the infection. 3 were colonized, but about 4 orders of magnitude lower than the no phage prophylaxis group. In the 24-hour prophylaxis group, they were all colonized, but again, a couple orders of magnitude lower than this group. And importantly, none of these animals showed any signs of disease, no watery diarrhea. We do see escape mutants amongst this Vibrio population, but again, as what we saw in the infant mouse model, we've yet to see an escape mutant, a Vibrio cholera come out of these animals that has escaped all 3 phages. Why is this? In vitro, we can put them through multiple passages and get mutants that can escape all 3 phages. I think we don't see it in the animals because of this principle I was telling you earlier. The phages are targeting essential virulence factors. And so I think that's a major determinant in Vibrio not being able to escape all these phages in the animal. So I mentioned that none of these animals had any symptoms. And indeed, if you look at body weights, there was not a significant loss in body weight. So this is body weight at the end of the experiment. These are just control infant rabbits that got neither phage nor Vibrio cholera. You'll note they lose a little bit of body weight because for this experiment, we take them away from the dams for the entire experiment because the mother will actually kill the infected infants. So the group that got no phage prophylaxis but that were challenged all lost significant body weight, and again, we had to euthanize all of these animals early on in the experiment. And then our 2 phage prophylaxis groups did not have a significant body weight loss compared to the control group. So it looks like this is working in these 2 animal models, and we're trying to move further with this into doing a clinical trial to see, in particular for this household transmission problem, can we protect the household contacts? So one thing we needed to do was show that this phage cocktail doesn't affect the normal gut microbiota. And so we chose to do this experiment in an adult mouse model, which have a stable microbiome. And so Lynn Cairns, the postdoc I mentioned previously, has been doing this work. And so to do this experiment, we have 3 groups of adult mice. This is a positive control for change in the gut microbiota, vancomycin treatment. This group gets 10 to the 8th of the phage cocktail, and then this group gets the heat-killed cocktail. And we actually had to repeat this experiment because the first time we did it, this group didn't get any treatment, and what we learned is that just giving them this oral gavage slightly changes their microbiota in the stool. So anyway, so that's our controls, heat-killed phage. And then we looked at, did 16S sequencing of the bacteria in the stool samples, and looked at any changes that we could see. And so what you can see is that the antibiotic-treated group dramatically changes their microbiota. This is a PCA plot, but the heat-killed treated animals and the phage-treated animals are all grouping in this tight cluster here, and we saw no significant change in their microbiota. Now, those that are familiar out there with these PCA plots, you can see most of the change here in the antibiotic group is along the x-axis here. And so what we did is if we remove this antibiotic-treated group and just focus in on this cluster here, again, we see no, so this is kind of a blow-up of this. Again, there's no pattern here. There's no change in the microbiota of the phage-treated ones compared to the heat-killed controls. And so this is kind of what we expected, but we had to show it. So Kim and others in my labs have shown that these phages don't kill other bacterial species, including even many closely related Vibrio species. And so we kind of predicted this, but we needed to do it for safety purposes. And so just to wrap up, So we've, in looking at cholera patient stool samples through these international collaborations, we've only been able to find 3 phages that appear to be fit to prey on Vibrio cholera in the human intestine. And so we've been using these in our phage cocktail. They all use receptors that are essential virulence factors, and this we think is the reason why it limits escape mutants occurring in humans, and probably reducing cholera pathogenesis. Certainly that's true in our animals, but remains to be shown in humans, but it's a good hypothesis. One of our phages can rapidly evolve to be resistant to this plea defense system that Kim talked about, and so that is a very interesting type of phage to have in a phage cocktail. And then this cocktail of the 3 phages can reduce infection and prevent cholera in these 2 animal models. And that the phage cocktail is specific for Vibrio cholera and doesn't affect their microbiota. And so right now we're working on how to produce the phage cocktail in huge amounts and keep it stable and have it be stable in oral vehicles that are FDA approved, and trying to get money to do a clinical trial. But we're excited about this and are moving forward on it. So let me just thank the former and present people doing this work. Kimberly Seed, of course, who did most of the work I talked about in the first half of my talk. This was started by Eric Nelson. I think I mentioned all the people in my lab working on this right now. I should also mention Dave Lazinski, a senior researcher in my lab, who has a hand in pretty much all of these experiments, or at least part of his brain in it. And as Kim mentioned, a lot of this work could not be done without access to cholera patient samples and this field work. And so I especially thank Firdousi Qadri, who I've collaborated with for many years, who's in Bangladesh. Stephen Calderwood, who's at Mass General in Boston, and Jason Harris have been part of these collaborations. And then the recent work in Haiti that Kim mentioned was done in collaboration with Jacques Bouncy at the National Lab and Luis Ivers, who has done a lot of work in Haiti as well. And I thank you for your attention.
Thank you so much, Andy, for a great talk. So we do the Q&A now. Maybe in the interest of time, we limit it for two questions and then take it to the reception, because we have food and drinks out there, and so people can, you'll be both around during the reception, right? All right. Okay. First question.
We're looking at that right now. The problem was to get the right clinical samples, so we needed to find, to get blood and stool samples from patients that also have a high titer of phage. And so we're collaborating now with Jason Harris, who now has those samples. And so we've just gotten serum, and we're now going to look. And so that could be a problem. We're hoping with oral tolerance and whatever that there won't be, but there might be. So we have done some, we haven't published this yet, but we've done some work in mice where we've generated high titer antisera against the phages. And for 2 of our phages, we get a high titer serum, but it does not neutralize the phages. But for one of the phages, ICP1, that high titer antisera will neutralize. And so there is the potential that that could be a problem, especially if we go to use this in Bangladesh, where these people may have seen these phages before and could have antibodies. Good question.
Another question, Jeremy?
The PEX-A system of ICP1 is very reminiscent of the conversion of phage lambda int into an excise by XIS. But I was just wondering if there's any evidence that one of the ways that ICP1 can fight back is by making an anti-PEX-A. So you don't excise the plea.
Right. Yeah, so as I mentioned, all of the ICP1 isolates have PEX-A conserved in the genome, so we haven't seen loss of it. We haven't specifically looked, I guess we haven't excluded that possibility that there's something anti-PEX-A to inhibit that and would compensate for, potentially the fitness defects of losing that protein. What I will say is that in all of the natural isolates that we've been able to test, they do excise plea in the lab. So there's no reason for us to think that there's something sort of more sneaky going on, but it's possible.
And a follow-up to Joe's question, is there any toll-like receptor in the intestine that recognizes phage coat proteins or tail proteins? Or
I'm not aware, I've never heard of any such thing. I don't know if there's any immunologists in the audience, but I mean, there's phages are being studied now for phage therapy purposes. They have been for many years, and when you give phages, they go in a lot of places in the body. But there's been no reports of them infecting cells or causing any ill effects.
Or anything. Yeah.
All right. Thank you very much. Also, maybe. All right, one more last question.
Yeah.
Okay.
This is for Andy. I was wondering, in the animals that received the phage cocktail, do you see a change in the phage populations over time?
You mean genetically?
Yeah, or just if you have a mixture of phages that are there, is there one that survives longer or better in the animals?
We haven't looked a lot at that, but we see amazing changes in the Vibrio cholera population, but we haven't seen changes in the phage population. But we haven't looked hard enough. They're probably, if you infect infant rabbits with Vibrio cholera and ICP2 phage, you can kind of reproduce what Kim showed in human stool samples. You get lots of escape mutants coming out. And it's possible that there's already some host-range phage mutants in there, but we have to look really hard to find them, and we haven't done that yet. But
Okay. Thank you very much. Also, thank you, the audience, for a very active discussion, and I welcome everyone at the reception.